Okay, we are going to, in this video, I'm going to teach you how to perform multiple sequence alignment and phylogenetic analysis. If you are not following, look at the last video, because we are using a particular sequence, consensus sequence. We queried and we got to this particular place where we are trying to identify the organism. We said for sure the organism is Bacillus, okay, but we are not so sure. We can see Cereus, we can see Tropicus, and so on. So what do you do in order to perform phylogenetic analysis? You first do what you call multiple sequence alignment before you will be able to do that. So to do that, we're going to continue. We look at this. We select the Bacillus, we select Cereus, select Tropicus, select Anthracis, then we select Subtilis. There is you can look for what you call an outgroup, different organisms, sequence of an organism, and also add. Okay, so but after you finish selecting here, you have to download the sequence. You click on this, you download faster complete sequence. When you click on faster complete sequence, your sequence should automatically download, and it will be in your download depending on how you set your download profile and all that. So for mine, it will be on download, and I can go to download and see that it has downloaded. If I come to this download, I will see that my sequence is downloaded here. You see the download as a notepad, to be at a notepad, as a notepad. So okay, look at that up here. Look at that, downloading. So it's completed. So I can go to my download and see it as a notepad, and I'm going to open it. When I open it, I see the sequences, and I'll add the query sequence, the one that we used, that consensus sequence. I'm going to add it into it because I want to align it with others. I want to align it and also do phylogenetic analysis. You know, I want to know, compare it with all these Bacillus, the Tropicals, and all that, to know how they are closely related. Which one is it my sequence more closely related to? That is the essence of phylogenetic. And which one is it distantly related to? So I will open this with a notepad. So it will open in the notepad. Okay, then look at it, see the first one, see the second one. If you go down, you see all of them that way. So I want to add my own, our sequence, consensus sequence. This is what we are using. If we copy it here and you add it to this, okay, let's look at it. You can edit this one, maybe to leave it as say only Bacillus. You can edit this one, leave it as only say Cereus. Okay, just shorten the name, you know, and all that. Okay, as only Tropicals, and actually it's normally better to maybe remove a common name, as this is Bacillus. Bacillus is none of them, so you remove Bacillus and leave it as Subtilis strain, and leave it in that way. It will be able to Tropicus here, you know, and this one Cereus. There shouldn't be something. Let's leave this as partition, then our consensus sequence. Then if we save this and close it, I normally use what is called cluster W. As I type in cluster W here, cluster W here, it will open to cluster W. I also use what you call cluster omega for multiple sequence alignment. I say cluster omega, you can see that, for multiple sequence alignment. So cluster W and cluster omega. I prefer cluster W. We'll see both of them. We'll see how they differ and all that. You see the difference between the two and why I said I choose one over another. Okay, it's opened, the cluster W. If we click on cluster W, it will open. If you click on cluster omega, it should also open. Cluster omega. So, but let me focus on our cluster W. Let's see how cluster W will help us to do the multiple sequence alignment. Okay. So cluster W, let's open it properly. Okay, let's see on the interface of cluster W. You see you can select proteins if it's protein sequence, but it is a DNA sequence. Later I will work on the protein sequence. So see, you can paste the sequences, you know, the way it is from the, we can copy it and paste it, or we choose the file. Remember, we saved it as a file, so I prefer we choose it as a file, you know. Then if we choose it as a file, we will go and to the download. Okay, go to download where it was downloaded and we saved it. Okay, all right, look at it. We open it, we load it here, look at it here, then we execute. So it will be running. While it's running, we can get to cluster omega, just look at it as it's another part of this. Look at what it is. So the same way, we select DNA here, we select DNA, okay, and you can copy the whole sequence the way it is, copy the whole sequence the way it is, or you upload it. Let's choose file here, also here, the way we did on cluster W, and also upload. Okay, let's look at both results and see why I choose one over another. It's not that, it's just a little difference in both of them. Okay, all right, let's see this. We choose our file here, that is on download. We click on download. That's where my own is. Yours could be in a different place. Our download is loading. Okay, look at it here. We click on that, then leave other parameters the way it is. Don't worry yourself about other parameters. Then click on submit. Let's go back to W. Let's see whether the result is out now. Alright, our cluster W results open. Let's see whether it's okay. Let's look at how the result is. Okay, look at it. You see, cluster W, these are consensus sequence, sequence 1, sequence 2, sequence 3, sequence 4, sequence 6. So it's, you see, it aligned. Consensus sequence about 1154. The other one is 600, the other one 1004, 1004. Then the sequence, it aligned 1 and 2. That means our consensus sequence and 2 is about 26%. The alignment score, align 1 and 3, 1 and 4 to 1 and 6. Then it will start aligning 2 and 3, 2 and 4, 2 and 5. That's how it will. I like to see this kind of alignment score. Okay. So at the end of the day, it will give us this type of alignment. Okay, to give us this type of alignment. So once you see all these type of stars, stars are telling you that in that particular column, all the bases are identical. That's what it means. So in this part, so what we do next is to build our phylogenetic tree. If you come here, select tree, you can use any of the trees. I will select a fast ML, but normally you can use field ML here to run your own field ML, and you click execute. You click execute. While it's executing, let's look at the other results of the omega. Well, it's still loading, and we'll wait a little bit for it to load, and let's see if we'll get our results faster depending on the network. That's so our result is loading. We have generated multiple sequence alignment result. We used it down to generate our phylogenetic tree. See, it's our phylogenetic tree is running. Okay, so the result is here. Okay, look at the results. So if we scroll down, you see the results. So look at the phylogenetic tree. So where is our consensus sequence? Oh, see our consensus sequence. So this is the root. From this root, this organism emerged, this other one emerged, this other one emerged. You see our organism here, consensus sequence on this different class. See this one, they are all in the same class. These ones are closely related sequences and so on. You see, it means that our sequence, okay, even though remember I just used one of it, I didn't use the it's just an example. The editing and all that is just for us to know. So when you see this kind of a thing, they are the same, means that it's a node. A node is where all of them emerged. Out of all the organisms emerged from here. One move this way, one move, and they come to this side. This one again here. You can change this if we click on without branch. You see, okay. I think it's better this way. That branch length. Let's look at it this way. So this is the node. This one moved this way. This moved this way, and came to this place. And this one branched here, this one branched here, and this place, this one branched here, this one branched here. These two are on the same branch. They are more closely related. They are on the same branch, are more closely related. So that's how you see that our organism is far on this. So if you want to look at it, you see the one that is more closely to our organism is this particular one. So in this way, you generate your phylogenetic. You can download it as PNG. If you download this thing, you can actually print it out. So in this way, you'll be able to build your phylogenetic tree. So in the next one, we look at protein. Here we look at nucleotide. In the next, we look at protein sequences, and we'll see how we'll be able to generate all these sequences, query our proteins,